Okay, so our group did the paper treating cattle with antibiotics affects greenhouse gas emissions and microbiota in dung beetles. Okay, so the relevancy of this study is that we need cows to eat, and there are 39 million cows in America. So those of which are composed in dairy and beef cows. Cows produce methane, which is bad for the environment and is actually much more potent than CO2 is. So it's a very potent greenhouse gas. Cows release between 70 and 120 kilograms of methane each year. So finding out ways to mitigate that is really important. Dung beetles are also involved in the study, and they're nutrient recyclers, which help break down the dung. So if it wasn't for dung beetles, there would be fields of poop and dead carcasses and all kinds of things because they're the primary nutrient recyclers in these environments. And antibiotics affect this process. In order for us to reach our food goals, we've given cows antibiotics to treat them and also to ensure the process of food is better. I guess that's kind of that. Three things that are looked at in this presentation are the gut microbiota, dung beetles and their effect, and also greenhouse gas emissions. So like I said earlier, methane is really bad for the environment. It's very potent. As this image is saying, it's 84 times more potent than CO2 is. And antibiotics do affect this, so we'll look into that greater as we go on the presentation.
So 10 cows were selected. Next. For the experimental methods of this paper, 10 cows were selected. Next slide. 5 of the cows were treated with tetracycline for a standard cycle. The other 5 cows were left as a control group. Next slide. Each of the cows, samples were taken from them 1 hour after the last administration of antibiotics. Next slide. And per sample, each cow sample was divided into 6 1-pound packs. So there's 6 1-pound packs per cow. Next slide. Each of the 1-pound packs were placed into mesh-covered containers. Packs 1 and 2 were tested for gas emissions and microbiota without the presence of beetles. Packs 3 and 4 were tested for gas emissions and microbiota with the presence of beetles. And packs 5 and 6 were used for sampling of the reproductive success of beetles and their overall fitness. Next slide. For packs 1 and 2, baseline emissions were taken from the soil, and emissions were also measured from the dung, and they were then stored in 95% alcohol solution to preserve the microbiota until DNA analysis could be done. Next slide. Packs 3 and 4 were tested the same way, except with the presence of beetles, and the beetles were homogenized to preserve the microbiota at 95% alcohol until DNA sampling could be done. Next slide. Packs 5 and 6, all of the larvae were counted and measured at varying intervals to measure if the antibiotics had any effect on the growth and reproductive success of the beetles. Next slide.
Okay, so now we go into the results of the experiment. And so what they found out after experimenting was that the dung and the beetles who eat the dung have very distinct microbial communities. These are the microbes found in the dung, and these are the ones in the beetles. So as you can see, they're very different. So now the question becomes what happens when you introduce antibiotics into the equation. And so these are those that weren't treated with antibiotics, and these are the ones that were. And so what that shows is that the antibiotics really did change the microbes found in the dung. So now what happens to the beetles who eat them, since they have very distinct microbes from the dung in the first place. So what they found was that those that ate dung that was treated by antibiotics and those that weren't also were very distinct, with the cream-colored circles being the ones that didn't consume the dung without being treated with antibiotics, and the blue ones were the ones that consumed the dung that was treated by antibiotics. Okay, and so how does this affect the beetles? And so a few theories they had was that it changed the nutritional quality of the dung, that it served as an inoculum, and it altered the microbiota of beetles as it did, as we saw in the previous chart, and it may be toxic since there not much testing has been done whether it serves as an antiparasitic. So that's maybe some reasons why the microbes were so different. And it also showed not to have an effect on beetle size, reproduction, and survival, not necessarily because it's not dangerous to the beetle, but more because it probably doesn't directly affect its growth and development, but could affect them in other ways. And finally, how it's so important in our lives today. Methane is a very potent greenhouse gas, and the beetles actually reduce the amount of methane released into the atmosphere. So these cream-colored bars are the ones that not treated by antibiotics, and these are the ones without beetles, and these are the ones with beetles. So you can see with the beetles present, not much methane is released in comparison to without beetles. But once you introduce antibiotics, the amount increases, very much so. And this chart is talking about the methanogens present. And so the methanogens found directly correlate to the amount of methane released into the atmosphere. And so those without beetles had more methanogens in dung versus with beetles. And then once you introduce antibiotics, that always trumps the without the antibiotics.
Okay, so there's a couple of major conclusions the researchers reached, and one of the first ones is the antibiotics directly affect the microbiota of both the dung and the dung beetles. And this is important because changing both the food source and the thing that eats the food source has far-reaching consequences for the rest of the ecology of the area. So other things that happen to eat the dung, or other things that eat the dung beetles, can pick up these trace amounts of antibiotics, and it could have a cascade effect that travels down the food chain. And there hasn't been any long-term research of this, so it could have really damaging consequences that aren't seen until it's too late. And another major part of this, it was shown that the microbiota of the dung beetles was changed, and it appeared that it didn't have any short-term effects, but there could be long-term effects that weren't part of the study. So another study maybe to go into that would be important to make sure that the dung beetle, which is super valuable to the ecology just because of how much waste it cleans up, it's important to make sure that it's not affected by the antibiotics as well. Next. And then the second major effect is the fact that by changing the microbiota of the dung, it allows the methanogen bacteria to outcompete all the other bacteria, and which therefore leads to methane being produced, extra methane being produced from the waste. And this is a huge consequence because there are about, as said before, there's 39 million cattle in the US. So anything that increases the amount of methane from the waste has dangerous consequences for the environment.